Hey, thanks a lot, Ben, for introductions. My name is Dan, and as was indicated, I've been at Oneonta for about 10 years now. We're really in like order of operations here on this slide today. Um, I should probably be towards the end if we're talking about the amount of work done, but we kind of wanted to take some separate bits and pieces here and talk about the work we've been doing for the last couple of years and tributaries to the Hudson River. Before we get going, I always forget people, and I'm bad at introductions as well. But just wanted to acknowledge, our partnerships with Riverkeeper, of course, Matt's here on the call, DEC, a number of others in the same office in which Billy works, and of course, WRI staff and a bunch of other collaborators whose logos aren't shown here on the first slide. And my favorite picture from the whole project had nothing to do with what we'll talk about today, because that's us over there electrofishing on one of our favorite stream reaches. Um, can you go ahead to the next slide there, Billy? Thanks. Again, I really am kind of peripheral to this work almost. And if you're from my era, you'll recognize this image instantly and realizing that I'm calling myself a talking head today. And so a quick outline of where we're heading with all of this so I keep myself on track and not get in the way of the good stuff. Cover some quick background in terms of what we set out to do, a little bit of why. Matt's really going to get into the applications here after we start to wrap up. Talk about the individual metrics that we chose to look at, and again, and why. Gross primary productivity or production, depending on what you like. So thinking about photosynthesis, oxygen, uh, and metabolic rates and streams, then moving up a level-ish, maybe two, depending on how you look at things, into stream macrobenthic invertebrates and how we might want to use those to characterize ecological function. And then finally starting to move up into trophic levels that I personally tend to think more about, things like river herring, and not part of this presentation, but now also eels as part of Billy's thesis. Billy will present results on those as we go. We'll try to wrap things up in a coherent fashion and kick things off here. You know, on the next slide, Billy? Okay, so a little bit of background. Most folks are probably familiar with the Hudson River drainage watershed. It's one of, the big main watersheds in our state. It's subject to some interstate management, in part because of the species that inhabit it. Are many of the ones that are commercially and culturally important are migratory in nature, move back and forth, and don't care a whole lot about provincial or municipal boundaries. So it's home to a large number of migratory fish species as well as resident fishes, of course. And it forms this massive network that we don't really think a lot about in terms of linearity, of over 3,500 river miles of tributaries, including the Mohawk River, the Upper Hudson, upstream of Troy Dam, and then many east and west tributaries as we move from the Adirondacks all the way down to the Battery in New York City. Um, Around 65, 70 tributaries, depending on how large the stream you're counting there. And just naturally, based on the geomorphology of the area, about half of those are actually blocked by natural stream features, things like waterfalls. And part of this is because of how the river channel was dug and left hanging kinds of tributaries there. And for that reason, it's got some features that are unlike many of the other estuaries and river systems in New York State. The remaining tributaries in the watershed are, for that reason, very critical as migratory fish spawning habitat, again, from kind of my perspective, right? But also for conveying nutrients in and out of the freshwater system back and forth to the ocean, et cetera, and all of those ecosystem services that are conferred to human beings as a result of these processes. Go ahead to the next one, Billy. And so The Hudson itself is a little bit more fjord-like than many of our other river systems in New York State. By that, a little more deep, straight, channelized. The estuary's got some really neat things happening as a result as well. And we've got this kind of conception that it's got a relatively productive biological main stem, right? We think about the main stem of the river and the estuary as really being this biologically productive powerhouse there. And to what degree the tributaries contribute to that or are impeded in that regard, we don't really have a great handle on. There are some decent historical data, and there are some newer ongoing efforts as well that some folks on the call might be familiar with. There's a lot of active restoration happening in the watershed. Matt certainly can speak to that better than I. And there are many other potential opportunities as we look, several years out down the road. It would be really nice to have some data to guide those opportunities, not necessarily for prioritization. Prioritization is great, and we want to be able to do that, of course, when it comes to understanding the benefit of potential restoration projects. But sometimes opportunities come along to do restoration that you might not have known about. And so part of what we're thinking about here with this study overarchingly is, can we get some basic metrics on the ground that could inform those efforts moving forward and say, might this be a worthwhile effort? Um, because of the somewhat abbreviated nature of many of the tributaries, again, this idea that we've got hanging tributaries in a fjord-like system, as well as a lot of natural morphological features that cause short stream lengths. Tributaries in the Hudson are often overlooked for funding when it comes to restoration projects. And part of the reason for that is we tend to think about how many river miles or how many river kilometers are going to be freed up for access to migratory fish. And if you're familiar with the large river restoration world and think about New England, think about the West Coast of the United States where large river restoration projects are underway. This is very often the status quo for measuring the impact of restoration. Would also be nice to be able to measure that biologically, though. And so that's kind of where we're coming from with all of this. And the idea here that we want to know is about how much do we get. This figure is showing, and it's just from like a Pearson textbook, but it's I think suitable for showing kind of global trends in productivity. And just one of the things that's worth pointing out here is you see the East Coast of North America, the Northwest Pacific, British Columbia, Vancouver, and then Northern Europe are really sort of hot spots for biological productivity in northern temperate zones of the world. So this is kind of relevant as well at a larger scale and may be able to inform, broader management perspectives. Go ahead to the next one, Billy. Cool. So really kind of what we're trying to get down to is what's in a river mile or really what's in the tributary, regardless of how many river miles long it is. And can we start to quantify just how productive these systems are in, maybe several different ways that may give us conflicting or agreeing perspectives. So we're going to talk about the same 10 streams or tributaries to the Hudson River. Those are shown over there in the map on the right. And the top left of that map is a little inset showing the watershed where it falls in New York State with the Upper Hudson and the Mohawk River included, of course. All of our work is happening downstream of Troy Dam or Federal Dam, whichever you're more familiar with. It's the same one. And the black dots in that give you an idea of the spread of the tributaries that we're looking at. And as we work our way down, we're looking at streams that you might be familiar with, Post and Kill, the Winans-Kill, Hannah Croix, Sauk Hill, Black Creek, Falk Hill, Quisseg, Cedar Pond Brook, Minneskiango, and Furnace Brook. And I still don't know where any of these are because I live in central New York. That's what Villia matter for. It's been a thing this whole time. They're divided about evenly east and west of Hudson. We tried to capture a range of human impacts. So some of these streams are highly urbanized. Some of them much less so, and even located on nature preserves, as is the case with Black Creek and Hannah Creek. Some of them have anthropogenic barriers, dams, on them, and then others have natural waterfalls that would impede, movement of nutrients or fishes, et cetera, potentially in different ways from dams as well. And so we kind of try to cast a pretty broad net despite having a limited number of systems here. Altogether, we're working with 20 sites. Each stream has a site that's downstream of the first barrier, whether it's a waterfall or a dam. And each stream has a site that's upstream of the first barrier, whether it's a waterfall or a dam. Goal here again is to get at characterizing the actual biological productivity and wrapping our heads around if we can think about that in terms of ecological value. So how are we going to do this? We're going to talk about stream metabolism. Really, what we're thinking about are oxygen cycles in the stream over a course of a number of days, and how we can use that. Some really standard macrobenthic invertebrate analysis to look at community dynamics there. And then we're actually going to use some recently quantified fish habitat that was part of the Atlantic States Marine Fisheries Commission 2024 stock assessment for river herring. And much of those data follow out of the work that Matt did with NOAA in developing the river herring habitat conservation plan. You can go ahead to the next slide, Billy. Great. So let's start out where I'm least familiar and thinking about photosynthesis. So we went into each of these sites, again, upstream and downstream of each of the dams, and with the initial intent of measuring stream primary production in stream, rates of photosynthesis or carbon fixation. They're all related, and you can measure one thing and get a lot of those things. So it's a really nifty set of tools. We had originally intended to use the classical light bottle, dark bottle method, and Billy having gone out and piloted that for us, so this is not going to work. And so we did a little bit of backpedaling. We ended up finding some additional funding to supplement on WRI funds and purchased a bunch of in situ hobo loggers so we could measure dissolved oxygen empirically at each of the sites and each of the streams, and then can derive gross primary production as a metric from the actual observed oxygen, dissolved oxygen concentrations in the water over time. Billy also went out and found this really awesome R package called Stream Metabolizer. That's awesome because it's Bayesian, circled in red there. And if we've met before, I could have a 3-hour conversation about anything with the B word in it. And so we went ahead and started setting up a study, looking at multiple days, oxygen concentrations throughout 24-hour cycles at fine scale increments. So we could iteratively estimate this really cool biological or ecological parameter. Go ahead to the next slide, Billy.
Oops.
And Billy, whenever you want to jump in on these chunks, you go ahead, okay?
Yeah, I can jump in.
Cool.
So what we did to get these stream metabolism estimates were we had these setups here with hobo loggers attached to either rebar or cinder blocks. And we did paired deployments of them upstream and downstream of the first barrier in all these tributaries. And they were deployed for at least 3 days. And then we retrieved them and then fit Bayesian state space models through the stream metabolizer package that takes into account observational error and process errors within the estimates. And these are some examples of the plots that you get from the stream metabolizer package. This is from the upstream site at Minisquiondo Creek down by Haverstraw Bay. And on the left plot there, you have 3 days of GPP and ER estimates with 95% credible intervals. And on the right, you have those modeled and observed DO. So top is DO milligrams per liter, middle is DO percent saturation, and the bottom is the rate of change of your DO. So the points there are the observed DO from the Hobo loggers, and the lines are the modeled DO from the Bayesian model. And it seems to underestimate the peaks there of DO, but overall we're pretty happy with the fit. And these are all of our GPP estimates from our tributaries. Each panel here is a different tributary. And on the y-axis, you have your GPP. On the x-axis is downstream and upstream sites. Downstream are in the light gray, and upstream is black here, and they all have 95% credible intervals. And what we saw was varied estimates among tributaries and between upstream and downstream sites, with generally higher upstream estimates above barriers. And also, interestingly, tributaries west of the Hudson River, which are Black Creek, Cedar Pond Brook, Anacroy Creek, Minneskiango, and Passaic Creek, were more productive than tributaries on the east of the Hudson River. So just to sum up what we did there, we had paired deployments upstream and downstream of the first barrier, whether it was natural or anthropogenic, and fit Bayesian models to get GPP and ecosystem respiration estimates. And these estimates are similar to historical work that was done in the Hudson with similar spatial patterns. So there was a study in the early 2000s by Thomas Bott, and he was working in some larger tributaries like the Esopus and Croton River, but they found west of the Hudson tributaries had higher GPP, and those watersheds were more open and less forested. And east of the Hudson River tributaries were smaller and more forested. So we're really interested to dig into some land use data in our study watersheds to see if we find any correlations like that. So to quantify some lower level consumers, we sampled for benthic macroinvertebrates. They're commonly used as biological indicators because they live most of their lives in water, and they're also they have varying sensitivities to pollution and also changes in habitat. And you can also break them into functional feeding groups, and that can tell you a lot about how the ecosystem functions and basal productivity. And this is a picture of a megaloptera or a dobson fly larva, which will be relevant in a couple of slides. So we used a server sampler, which is pictured there. It has a metal frame that you embed into the stream bed, and then a net attached to it, and you face the opening of the net upstream, so water is flowing into it. And we sampled at a riffle, a run, and a pool habitat at both upstream and downstream sites in all our tributaries. And you take up the large cobble first and kind of clean off the cobble, and the macroinvertebrates flow into the net. And then once you get through all the larger cobble, you agitate the smaller sediment. And we went to a depth of 4 inches just to get all the macroinvertebrates that are stuck in the finer sediment. And we identified and enumerated all of them to get abundance and diversity. And for biomass, we first dried them in a drying oven at 60 degrees Celsius for 24 hours, and then weighed them to get dry weight. And then we burned them in a muffle furnace at 500 degrees Celsius for an hour and measured them to get ash-free dry weight. So this is a plot of the numerical abundance. So each panel here is a different habitat type. The y-axis is raw count numbers, and The x-axis is our tributaries, with the dark gray being downstream sites and black being upstream sites. That'll be consistent for the next couple of figures. And overall, we see higher abundance in riffles and lowest abundance in pools. And this will be a trend for pretty much all of our macroinvertebrate metrics. So this is biomass, looking at dry weight and ash-free dry weight. We see the same thing, same pattern as abundance, with riffle being the highest and pool being the lowest. So some of these large peaks here are those Dobson fly larvae, because they're large and they weigh a lot and they're really creepy. So they kind of skew the results a bit. And in the bottom right, in that run habitat downstream Sawkill, the ash-free dry weight's kind of off the charts there, because we caught a snail there. So that's a snail shell. So family level richness is a measurement of the unique species that you're seeing. And same trend as abundance and biomass, a little bit less variable, but you have riffle with the highest diversity, run a little bit less, and pool less than run. This figure is kind of crazy, so bear with me here. We have the functional feeding groups for all the habitats within sites in each tributary. So CF and CG are your collector filters and collector gatherers, and they're mainly feeding on the fine particulate matter. Predators are eating other aquatic insects. That's PR. SC is scrapers, and they're kind of scraping wood and rocks and aquatic veg of periphyton. And the SH are shredders, and they're eating more coarse particulate matter like leaf litter. So there's a lot to digest here, and we're still working on that. But just surface level look, it kind of seems like the collectors and scrapers are dominant in all of our habitats across tributaries. So to sum up the macroinvertebrate stuff, all of our metrics, abundance, family level richness, and biomass, kind of follow the similar trend where everything's higher in the riffles and lowest in the pools. But environment and vertebrate biomass is higher upstream of dams and waterfalls. And digging into those functional feeding group patterns will be interesting. It'd be interesting to take those same land use data and apply it to our invertebrate samples and see if there's any correlations there. So finally, to quantify our higher level consumers, we looked at river herring production potential. So alewife and blueback herring are combined as river herring. They're anadromous, which means they come into the streams to spawn, and that happens every spring, where hundreds of thousands, if not millions, come in to the Hudson River and their tributaries, bringing with them like an influx of marine nutrients and productivity. And the latest stock assessment, benchmark stock assessment in 2024, had newly developed habitat models, which we used in combination with these stochastic population models developed by Dan Stich and colleagues to get estimates of blueback herring production potential in all our study tributaries. And that's in the Anadrafish R package. So how we did that was we had two different scenarios, a no passage scenario and a no dam scenario. So a no dam scenario takes all of your blueback herring habitat up to the first dam and runs the population model just on that habitat. So that's pretty much the status quo, assuming that river herring can't get above those dams. And the no dam scenario takes the blueback herring habitat above those dams and includes them into the population model. So it's like a hypothetical scenario, a world with no dams where river herring have access to all the known blueback habitat. What could the tributaries support? So the results for these vary among tributaries. Uh, Tributaries where the first barrier is a waterfall didn't get a no dam scenario because even if you removed all the dams on those tributaries, their first barrier is a waterfall, so it makes no difference. What they're able to access today is what they're going to access forever. So tributaries with a no dam scenario, Cedar Pond Brook, Furnace Brook, Minneskiango Creek and Passaic Creek all had increases in available habitat, and that leads to an increase in production potential for blueback herring. All right, Dan, you want to take us home?
Sure. So kind of like pull it all back together, big picture. We are seeing differences in primary production among the tributaries between the sites located upstream or downstream of the first barriers, specifically dams as well, in these systems. That's good news. That means there's variability there that could be leveraged potentially for restoration, right? Um, Not all those trends are exactly what we expected. As Billy had indicated, it seems like we actually have higher productivity going on upstream of uh, both anthropogenic and natural barriers. So this may be more of a hydrogeological phenomenon than it is necessarily an impact of anthropogenic dams. However, we don't know what that looks like either. If you remove one of those anthropogenic barriers, does the rest of the river kind of even out in that way? So certainly there's productivity being locked up behind these barriers. We saw that recapitulated in the invert abundance, diversity, biomass, which is really intriguing there. So it suggests that there is potential to kind of reconnect those systems biologically with regard to both primary production as well as the stream invertebrate communities. And then, of course, we see changes in the river herring population because we imposed those through a simulation model. But we now have a bit of a better understanding of what the magnitude of increase could look like in those river herring populations. This really starts to move the ball down the road from thinking about how many miles of tributary are open. And in some cases, we only have a few miles of tributary. And it moves us from thinking about miles of tributary to thinking about changes in stream metabolic rates, changes in stream invertebrate communities, and then, of course, changes, at least relative magnitude changes in river herring abundance and population sizes. And these get us really neat, direct measures of those kinds of things that are not necessarily captured by measuring how long a reach of river is. Go ahead to the next one, Billy. I think it's just yeah, next step. We've got more coming. We've as long as we were driving up and down the Hudson River Valley this summer, we decided to stop and catch a few thousand eels in collaboration with DC, and went and actually sampled all of these sites for American eel using three-pass depletion methods as well as environmental DNA. So Billy's got a whole nother chapter of the thesis to go with some more empirical data. This will really give us a nice perspective on a top level consumer that eats all of the things that we were talking about. Well, it doesn't eat oxygen. That would be weird. But it eats all the other things that we were talking about today. And they also, rather than having babies in these tributaries, this is where they go to grow old, right? And they might live there 5 years if they're a small male, up to 20, sometimes 30, in a crazy case, 40 years if they're a large, reproductively important female. So this is going to give us one more lens to look through as we start to think about this and compare those numbers of eels upstream and downstream of the anthropogenic and the natural barriers. And just kind of spoiler alerts, eels have a little bit easier time moving around waterfalls than river herring do as well. So I suspect we're in for some interesting stuff going on there as well. And then the last slide is thank yous. So, always good to say thanks. Of course, WRI provided the sort of initial funding that got this all off the ground. Epley Foundation was really awesome, and they about matched the funding there so that we could cover time for people in the field and so that we could get more supplies as we were contingency planning at the last minute because our bottle method for dissolved oxygen didn't work. The SUNY Oneonta Biological Field Station provided us with many of the other supplies that we needed and covered. Some of my time, of course, Riverkeeper, and Riverkeeper sent out an army of volunteers as well throughout the summer. DC also many folks from Billy's office and others, as well as volunteers from people in our own personal lives to graduate students at SUNY Oneonta and folks who work with all of us in our very different locales.
I also I wanted to thank Dr. Jeff Helville at SUNY Oneonta for identifying all 1,600 of our macroinvertebrates. We're a bunch of stupid fish biologists that don't know anything about bugs, so it was really clutch of him to do that.
Yeah, and speaking of Riverkeeper, Matt, do you want to tie us all together with, what this research might mean, and just give us a little bit of, your insights and how this type of research can impact, management decisions and, your kind of world.
Yeah, for sure. And yeah, thanks to Dan and Billy for great job on the talk, and obviously great job pulling all this research together. And thanks, Ben, for having us talking today. Dan touched on it a little bit in the beginning on some of the practical uses that this research will hopefully be used for. I like to think of it in kind of three separate bins. The first being for Riverkeeper's work, it really helps us kind of focus and narrow and prioritize where it's most beneficial to start doing restoration work and start doing dam removals and things like that. The second one, it really helps us go out and get funding as well for these restoration projects, which are not cheap at all. There's engineering costs and construction costs and things like that. So, and not just for Riverkeeper, but for other practitioners throughout New York State and the Northeast and the Hudson Valley, they can hopefully use this to advocate for funds to do restoration work on tributaries and to reconnect tributaries through dam removal and culvert replacement, things like that. And another piece to that puzzle that Dan mentioned earlier, if you remember that map of the primary production across, particularly if you zoom in on the U.S., this region seems to miss out on quite a bit of federal funding from time to time, because our tributaries are so like serially dammed that taking one out only gives you about maybe a quarter stream mile in some cases, whereas out west you're taking out a dam and opening up like 20, 50 miles. But if you look at that productivity map you see in this Northeast area, we're in a pretty productive area in terms of we got our forests here and dropping leaves into the water for all these shredding macroinvertebrates to eat. So that quarter mile is really productive here if we're able to open it up and restore access there. And then the last bucket of usefulness is this wealth of data. both in terms of kind of pre-removal data. So as if we're able to then take out one of these dams that we talked about today, we have a really great set of data to then compare to if we go and reproduce this study after the dam's gone, and really get to see the benefits of removal, which then ties back into the other stuff that I mentioned about funding. If you're able to see those benefits, you can more easily get more funding in the future. And then the last piece of data is, of course, for to bring it all back to fish is great for fisheries managers to know, for species like river herring and American eel that are at historic lows, to better understand how to bring them back.